Good morning, everyone. Thank you for joining so early today. This morning we have Dr. Janet Wittes with us to speak on the oversight of DSMBs in clinical trials. I'm sure that no introduction will suffice to capture her many accomplishments over the years, some of which involved work with many of you, but I'll make an attempt. So Dr. Wittes began her educational journey at Radcliffe with a major in math, after which she moved on to Harvard to pursue her master's and PhD in statistics. Her career has spanned academia, government, and private industry, with the greatest chunk spent as president at StatCollab in Washington, D.C., where her company was awarded countless government and industry contracts for a variety of trials. Recently, Dr. Wittes has stepped out on her own to found her own consulting firm, where she continues to actively advise on the design and conduct of clinical studies. Most pertinent for today, however, Dr. Wittes has served on 57 different data and safety monitoring committees. So I'm sure there's plenty that we can learn from her today, and I'm sure she'll be willing to take some of our questions as well for some of our struggles. Janet, whenever you're ready, we're ready to have you.
Okay, well, thank you so much, Teresa, and I'm really glad to be here, and thank you all for being here. So I want to start with a very personal story. Imagine almost 75 years ago, when I was then almost 7 years old, and we were living in Brooklyn, which was then, if it had been if the 5 boroughs of New York had been separate cities, it would have been the largest city in the country. And it was very urban. And we were moving to New Milford, Connecticut, which was a very small town at that time and very rural. And my mother was in graduate school. My father believed that smart women should go to graduate school and get PhDs, which was very unusual at the time. And we were driving. My father was driving in the front seat. My brother, who was then 3.5 years old, and I was, as I say, almost 7, sitting in the back. We drove all the way to the country, stopped in this dead-end road by a big, what seemed to me a huge house, because remember, we were living in Brooklyn in an apartment. And my father went out to look at the house, and he said to the two of us, don't come in, don't leave the car. And he left. And my brother, of course, opened the door and ran in with my father and went into the house. And I was sitting there steaming. And when they came back, I said, Daddy, that's not fair. Johnny got to see the house and I didn't, and I was obedient. And my father very calmly said, Janet, it's time for you to learn when not to listen to the rules. And that was, whoops, I can't. Well, there it is. And that was one of the most important lessons I ever got in my life, that there are rules that we should have. So originally I was going to have a talk, should DSMBs overrule their boundaries? But given what I learned as an almost 7-year-old, the real question is, when should DSMBs overrule their boundaries? Because the context of overruling a rule that one has, when one should overrule a rule that one has, depends on the context. And there, as a 6-year-old, the context was running out into the lawn, seeing a new house. The rule of staying in the car made no sense. So I want to remind you first about the structure of DSMB. That's what we're going to be talking about. The structure of a trial. There's a sponsor who's responsible for the trial and reports to the regulatory agency. And there's a steering committee, which is it and the sponsor together are the intellectual hubs of the study. There's the independent data monitoring committee, which I'll sometimes call data monitoring committees. I'll sometimes call data safety monitor. I'll give them different names, but they all mean the same to me. And then there's a group that reports to that data monitoring committee. And I call that the independent statistical reporting group. And we were going to be talking about that yellow box. today. But remember that there are lots of other components, and I know that all of you are involved in some very complicated trials, so that you know that there's the data management committee, and sometimes it's you guys, sometimes it's a sponsor, sometimes it's a CRO. They're the clinical center, they're the participants who are actually in the trial, the patients in the trial, they're the central units, they're the IRBs. And it's important to remember, I'm sure all of you know, but it's very surprising to me how many people don't know that the IRB is different from the DMC. The IRB has the overall, it represents the units in the study and looking at the ethics, but doesn't have access to the data during the trial. It's only that yellow box that has access to the data during the trial. And I'm going to be speaking mainly about phase 3 or phase 4 trials, mainly about blinded trials. But some of what I say has to do with other trials as well. So if you think about the structure, it is a complex system. And Herb Simon won the Nobel Prize in Economics in 1978 for, and this is the quotation, for his pioneering research into the decision-making process within economic organizations. And we can cross out the word economic and change it to clinical trial, but the crucial point there, the crucial words are decision-making. And then he says, a system that can be analyzed into many components, having relatively many relations among them, so that the behavior of each component depends on the behavior of others. And I can think of no better description of a clinical trial as Herbert Simon's description of a complex system. So I'm going to have this, the outline of this talk is going to be in 3 parts. First, I'm going to read this part very fast. This is what you already know, but I'm going to bring this up because so many people, after they leave school, seem to forget or ignore the principle they've learned or even knew before they went to college or graduate school. So that's the first part. Then a very quick review of stopping for safety, futility, and efficacy, stopping a trial. And it's quick because I'm assuming all of you kind of know what the rules are, but I just want to get us into the same framework, and then when to break the rules. So here's what I'm sure you all know, but a data monitoring committee needs good reports to make good decisions. And I can say, having sat on some of the DMCs that you guys prepare for, your reports are really good. Don't forget how you did them, because reports should be clear, complete, and comprehensive. And we wrote a paper about that. We should have added concise in our titles, but they should be a report that somebody can look at. And remember the Herb Simon, the purpose of a DSMB is to make do decision-making during the course of a trial. So I want to give you some lousy examples so that you'll never do this, but these are real examples of what people have given. When I sat on, I didn't realize, Teresa, that I had 57 of them. When I have sat on DSMBs, I've had some terrible reports, and these are from people who had really good training, just like all of you have had really good training, and then they go out in the real world and they do these awful things. Here is a report I received. It was an email with a zip file, and this is all that, on the left hand, where are these RTFs? No titles, no nothing. This I'm supposed to click and click and click and click and figure out what the report is. That's one report. Here's another one, stack of binders. They come in the mail, these binders, no table of contents. And when you look at the binders, it just says table 1.1, table 1.2, no description. Again, these are from people who've gone to really good schools, who knew what was better, who couldn't have been trained like this, and this is what they did when they grew up. Here's another one. Here's a safety report. And these are not fake, these are real. So here's the report, screening, randomization, and then it's going to show data over time. And then there's, in case you don't know that 0 over the denominator is 0%, it tells you that it's not only 0, but the 0.00%. So if you kind of look, and the items are out of order because there's early termination, which may have occurred early, there's unscheduled visits, and there's 150 pages of this. Every so often, you'll see a 1 or a 2. And if that's not enough, there was change from baseline. When the baseline was missing, they call them zeros, so they'd have baseline variables, lots and lots of decimal places, p-values to 3 and 4 significant, and so forth and so on. So don't do that. And when you're in a situation where that's what these are the kinds of reports that you're told to do, please refuse, break your rules. So, and finally, remember that a final report for a study needs to be accurate, but a data monitoring report needs to be timely. Many reporting groups insist on clean data, but that means because data emerged from the study often late, don't reflect current data. And to be up to date, some of the data will be dirty, and but the DSMB must know what data are not yet reliable. Dirty doesn't mean sloppy. Remember that women don't have prostate glands. So if you're looking at prostate issues, don't include the women in the denominator. Don't clutter up solid tumors with benign polyps. And think about the linkage between adverse events, serious adverse events, and laboratories. Again, all part of a complex system. And in general, think. If you see potassium, remember normal potassiums range between about 3.5 and 5. Normal sodiums range between 135 and 145. If someone has a potassium of 140 and a sodium of 4, switch, right? You don't have to. I was involved in a study once where there were hundreds of these switches, and the company went for each one and queried whether these should have been switched. Here's a wonderful story from, it's funny, I can't read my slides because sorry. Well, anyhow, the British Airways didn't let a musician flying with her cello didn't let the cello on the plane because it didn't have a visa. The cello had a name, Chuck Cello, but the rule was that anyone or anything with a name had to have a visa, and so she couldn't travel with her cello. So if the data monitoring report, if the DSMB receives inadequate report, don't expect sensible recommendations. So here is a summary of what a nice report should have information, so that think about, as the DSMB is sitting there, having not thought about the study, maybe for 6 months, you've been involved in the study every day, it needs to be reminded of what the study is about and what the data are showing. Okay, with that as an introduction, and I'm sure you all know it, but I just want to warn you that as you leave the nice halls of the university, there will be people pressing you to do these awful reports, do good ones. So what are we talking about when we talk about stopping boundaries, or are they guidelines? And I'm going to talk about 4 types of reasons for early stopping. The first is the study isn't going anywhere. The recruitment's too slow, the follow-up is too lousy, the data are too inaccurate. And there are, in those cases, you may want to stop the study just because, hey, we're doing a study, but it's not showing, we're very unlikely to get any reasonable conclusion. So here's a difficult case where this was lousy reporting, and the DMC had been looking at the data for about 3 years. They were meeting every 6 months for about 3 years, and they saw no difference in the treated and control. Usually you can see, you look at the safety, you can see something in the safety. There's usually some difference in the treatment and control, even if there's no benefit. In this case, the disposition was the same, so the dropouts were the same, the amount of follow-up was the same, the safety was identical, almost a little bit of variation, no difference in efficacy. And the committee kept on asking the reporting statistician, my role was to look at this after the study to see what had happened. And at one point, the chairman was really, about 2.5 years in, was really upset and said, "Are you dead sure you have the right code?" And the reporting statistician said, "Yes, everything was audited. This is the right code." The data monitoring committee thought very hard about recommending stopping, because it said there's something very wrong with this study, and we don't think you're going to be able to conclude anything from it, because we don't believe that the data we're seeing are the correct data. But it didn't stop. And why didn't it stop? Well, there were 2 trials, actually. The trial that it was looking at that had these troubles, and another trial in another region. This was a vaccine trial. And as you know, that vaccine trials tend to have a Northern Hemisphere trial and a Southern Hemisphere trial, especially if it's flu. This wasn't flu, but where the disease comes, it cycles in time. And so the Northern Hemisphere gets the disease in its winter, and the Southern Hemisphere gets its disease in its winter. So they had these 2 trials in 2 different regions. And it was the same vaccine and the same disease. And the other trial was showing clear evidence of benefit. So the DNC said, well, let's just let this trial go on, and then at the end of the day, we'll understand what happened. Had there been no other matching trial, they probably would have stopped. At the end of the trial, the company found benefit in both trials, and some adverse events in both trials, because almost every drug has some adverse events. And in fact, the reporting group had been using the wrong code all along. It had been using a mix-up code, and the auditing was an audit that, this is not the time to discuss what the audit was, but just because something is audited doesn't mean it's audited correctly. So that's a case of a difficult case where the trial was going nowhere, but luckily they didn't stop it. And those, I would say, low quality reports. Now here's some cases, difficult cases with high quality reports. And I'm going to be talking about now safety concerns. Safety data must be coming in quickly. You need to think about what would lead a data monitoring committee to recommend a change in the trial on the basis of safety. Too many deaths, too many serious adverse events. It has to be thinking collectively that the balance of risk and benefit favors benefit, even if the efficacy data are not available. Now, why would the efficacy data not be available? Because sometimes the adverse events, you take the drug and the adverse event can happen quickly. The benefit may be not apparent for a long time, or the benefit may not be apparent if it's a preventive trial. It's not apparent to any individual. It's only apparent when you look at the 2 groups. And the data for safety are often collected in what's called a regulatory database. It comes in much, much faster than the data from the CRFs. it avoids the clinical database. So the safety data comes in quickly, and the data monitoring committee has to think about, okay, I'm seeing these bad things happening, but are they going to be, is the balance of risk and benefit going to turn out to be beneficial when all the data are in? Usually, and that's all I'm going to say on safety data. Usually there are no bounds for safety data unless the trial is specifically a trial aimed to look at safety. But this is usually the judgment of the data monitoring committee, judging that the adverse events are either currently, are likely to be worse, to have at the end of the trial an adverse benefit risk assessment. And then there's the futility, or the other way of thinking about futility, lack of evidence of benefit. Very often, a futility bound is clearly laid out. Sometimes the futility bound will be what's called mandatory. And I would say if you have a mandatory futility bound in your study, if you're on a city on a DMC and the bound is mandatory, and your charter, refuse that. So here's a couple of examples of trials where there was a very clear futility bound, and the DSMB said, no, we're not stopping. This is a phase 3 trial, and I wish I could give you the study and so forth. It's still not public. This is a phase 3 trial of cell therapy and heart failure. So this is a pretty new kind of treatment with great promise for the future, but not a lot of experience. It was unblinded because the cell therapy was, it was just very difficult to blind. It was randomized 1-to-1, about 75 in a group. Now, remember, most heart failure trials, well, I shouldn't say most, many heart failure trials that are looking to show benefit in a new drug are thousands of people. When we're talking about cell therapy, these are cell and gene therapy, these tend to be very small trials. Here, the primary and secondary endpoints were defined very clearly. And the study was continuing, and the DSMB was meeting about every 6 months. And there was no way that either the primary or the secondary would show benefit. There was essentially, on the endpoints, there was essentially no difference between those who received the cell therapy and those who didn't. The futility rule was very definite. It said to the DMC, you have to stop if you see no evidence of benefit on either. And the DMC saw clearly no evidence of benefit. But there was apparent activity of the cell therapy. Everyone had been randomized already. So there wasn't, so whatever was going to happen to these patients had already happened. And it kind of looked like benefit early on one of the measures of left ventricular function. And the data monitoring committee said, okay, we know we're supposed to stop, but let's hold our breath and let this study continue. It's not going to hurt the patients that are already there because there's nobody else being randomized, and their treatment is going to be the same whether we stop the study or not. And the only one that's going to hurt is the company, but that's not our responsibility. Our responsibility is to the science and to the participants. So the study went on for another 2 years. At the end, there was clear benefit on left ventricular function. The sponsor was extremely grateful. I mean, we had a meeting of the DMC and the sponsor showing them what had happened and why the decision had been made to continue. And what they said was, well, we didn't think it was going to have effect on left ventricular function, so we didn't consider that an endpoint. And if you had stopped the study, we would have stopped development of the product. But now there was a path forward. So everybody, the DMC, which had been holding its breath, breathed a sigh of relief and thought, okay, we did a good thing. This was good. But now I want to give, so here's an example. That's an example of let's continue when we shouldn't have, and that was a good thing. So here's another example. Let's continue when we shouldn't have, but it wasn't such a good thing. Study number 2, 52-week continuous endpoint. And the interim analysis, there was an early surrogate that was highly correlated with outcome. And again, I can't tell you the details of this. You just have to believe me. And they said, the charter said, if you see no evidence of benefit, recommend stopping this study. And they had a very specific criterion: stop if the conditional power for the current trend is less than 30%. So that is, if the current trend continues, but remember, this is a current trend on the early surrogate. There's no data on the outcome, on the 52-week outcome, then you should stop. But the charter said, but this isn't a mandatory stopping. Use your judgment. So the committee saw absolutely no benefit on the surrogate. The conditional power was way under 30%, but the confidence intervals were very wide. And so they could have been benefit in the surrogate, and they didn't understand, or we didn't understand, the relationship between the surrogate and the 52-week continuous outcome. Nobody had 52-week data. Recruitment was still ongoing. So this was more complicated in some sense than the previous one, because now people were entering a study where we had evidence that it wasn't working, at least on the early surrogate. But what the Data Safety Monitoring Board said, look, if we recommend stopping and there's a delayed benefit, nobody will ever know, and we'll kill the product. So the recommendation, and this was a heavy recommendation, was continue. This was not as clear to the committee as the previous one, where there was nobody still left to randomize. So what happened at the next interim? The 52-week continuous endpoint, still no benefit on the surrogate, now with many more patients. The conditional power at 52 weeks for the continuous endpoint was less than 10%. So there was and the secondaries were all in the wrong direction, not strongly in the wrong direction, but in the wrong direction. And so the committee asked ourselves, was it right what we did at the first interim? Given what we know happened at the second, it would have been better to have stopped at the first, but would that have been the right decision? And we were all sort of comfortable with, well, we did what seemed to be the right thing given the data that we had. But I don't want to give you the impression that every time you break your rules, that's the right thing to do. So, and then let's talk a little bit about stopping for overwhelming evidence of efficacy. And I want to remind you that usually when there are bounds or guidelines set for efficacy, there's been a huge amount of work setting those guidelines. These are not just, oh, we're going to do a boundary, and if you cross the boundary, stop. These are a lot of work deciding when the looks are going to have, what kind of boundary to have, what kind of criteria, what should the DMC look at, all kinds of stuff. So a DMC doesn't want to willy-nilly overrule the guidelines. So when you're looking at the efficacy analysis, but again, remember that if a DMC recommends stopping a trial and the trial stops, it can't restart. So stopping is a very big decision. So you look at the primary and the secondaries. If you're thinking about stopping, you want to look at the subgroups of interest. Is there going to be something in one of the important subgroups that are going to make people say, oh, they shouldn't have stopped? What about adjusting for covariates? Anything else that could influence the recommendations of the DMC? Because, as I just said, a stop trial cannot be restarted. There are occasions when it can, but basically it can't. So here's the first trial that I was involved in where we disobeyed the boundary, like my brother running out of the car when we were told not to. This was the CURE study. It was published in the New England Journal in 2001. There were 12,562 patients with unstable angina, a typical sample size for a cardiovascular trial, randomized 1-to-1 to receive clopidogrel plus aspirin or placebo plus aspirin, so blinded. Patients followed 3 to 12 months. And so this is the results from the published in the New England Journal. And the first primary outcome, there was a complicated trial, so there were more than one primary outcome, but the first one was death from cardiovascular causes, non-fatal MI or non-fatal stroke. And clopidogrel, the rate was the percentage was 9.3%, and placebo 11.4%. The relative risk was 0.8, confidence interval 0.72 to 0.9, and the p-value less than 0.001. So obviously, if the p-value at the end was less than 0.001, and remember the New England Journal doesn't let you write down anything more precise than 0.001, it was much smaller than that, it must have been small during the study. So here's what the final paper says. The Data and Safety Monitoring Board monitored the incidence of the primary outcome to determine the benefit of clopidogrel using a modified Hay-Biddle-Peto boundary of 4 standard deviations in the first half of the study and 3 standard deviations in the second. So we were watching it. You the p-value had to be really, really tiny in the first half and pretty tiny in the second half in order for us to recommend stopping. Furthermore, if that's not strong enough, the boundary had to be exceeded at 2 or more consecutive time points, at least 3 months apart, for the board to consider terminating the study early. Now, if you look at what these rules are, think of what we must have been thinking during the DMC. This is really clearly coming from a sponsor or from an investigator who doesn't want you to stop the study. They want you to be really sure that there's benefit. And then it says, this is all from the New England Journal, there were 2 formal interim assessments performed at the times when approximately 1/3 and 2/3 of the expected events had occurred. And here was the committee. George Weiss was the chair, and we all knew each other very well. We had all been sitting on committees before. We knew each other, and we knew the PI, who was Salim Youssef, and we knew he didn't like people to stop. So, and this is what we were seeing from the very beginning. This is the paper, this is not the actual data that we saw at the DSMC meetings, but this is the data at the end. You can see what we were seeing was essentially nothing for the first month, and then the curve split apart very early. And the data clearly crossed at the second interim analysis, and the DMC, the New England Journal says, despite the fact that the preset boundary indicating efficacy had been crossed by the time of the second interim analysis, the board recommended that the trial continue until its planned end. And then with the crucial part, in order to define more clearly whether the risks of major bleeding episodes could offset the benefits of therapy. Now, I remember very clearly the meeting we were sitting in the hotel room at the Sheraton Hotel at the Toronto airport, sitting around a table. seeing the very, very clear evidence of benefit on a really important endpoint. I mean, this isn't a trivial thing. This is cardiovascular death, MI, and stroke. And yet there was some very severe intracranial bleeds. And we said, we got to recommend, we can't let this study stop, because in order to use this drug, people have to understand the risks of major bleeding. And we were extremely nervous about not recommending stopping. I mean, each one of us said, will you come visit us in prison if we get sued? Because we really recognized that this was a heavy decision that could be life-changing for some people. And this was the first time I realized how much I don't like voting in a DMC. And we didn't vote. The chair, George, went around to each of us and made each of us say why we were thinking what we were thinking. And we didn't make our decision until there was a consensus, a really comfortable, well, it wasn't comfortable, a real consensus. It wasn't comfortable because we were nervous that this was that we would be in trouble and that this was a bad decision. But we felt really strongly that you had to understand the bleeding. So, and at the end, the investigators were very glad that we continued. We had recommended continuing, and they actually made in further charters, they gave even more latitude to the DSMB than they had given to us. Here's another example, the Rewind trial. There were 9,901 participants. It's too bad that extra one is there, but she was there. Randomized 1-to-1 to dulaglutide, that's naturalistic or placebo, mean age 66.2 years. And this is to look at among people with diabetes, cardiovascular events, and almost half of the population were women. And this is again another trial that came out of the Population Health Center at Hamilton. So they had a pre-specified Hay-Biddle-Pedo boundary, not quite as extreme a boundary as the one in for cure, but pretty extreme. And at the last interim analysis, the trajectory of the primary outcome had crossed the pre-specified Hay-Biddle-Pedo boundary. In spite of that, we recommended continuing. Let me see if I have, and you'll see in the Lancet the article. And so why didn't we recommend stopping? Well, the effect was all in the males. So there was essentially nothing in the women. And we sat there, again, I remember this was an airport hotel in Miami, and I was chairing this committee. And when I had looked at the data before coming, I thought to myself, everybody else is going to want to recommend stopping this trial, but I don't because of the women. And I'm going to be the only one that says this, and I'm going to have to argue, and it's going to be really uncomfortable. Charlie Hennekins was sitting to my left, so I was going to go around the table, and Charlie started, and I didn't say anything because I was the chair, so I don't have to say anything first. And Charlie said, look, none of you is going to agree with me, but I don't think we should stop. There's no effect in women, and this is going to be really confusing to people. And I know the rest of you are going to disagree. And as he was saying this, you could see everybody's face, the sense of great relief. And it turned out everybody had the same feeling that, first of all, that they didn't want to stop. They knew that everybody else would want to stop. And it was because of the women. So what happened at the end of the day, the hazard ratio was 0.88, confidence interval barely below 1, p-value of 0.6. And what was interesting, so by the end of the trial, there was some effect in women, not very much, but it wasn't nothing the way it had been when we were looking at the data. And interestingly, the FDA label, we don't know what was discussed at the FDA, but they must have said, we don't believe the sex result. And it reports the rewind data overall, does not give in the label, no results by sex. The label is for adults. And what we as the committee thought, if we had stopped this study early without a hint of a benefit on women, it was just almost, the hazard ratio was like 1, there probably would have been a statement that there's no evidence of benefit in women. But by letting it go on, you could end up with a label for adults. So I want to give you a couple of other difficult problems. Here's one with no interim analysis plan for efficacy. And the FDA had said, and again, I just wrote to the person who's written the paper about this DSMB, but it hasn't been published, so I cannot tell you what it was. But this was a very, very serious neurologic disease. And the FDA said, no matter what you see, do not stop the study early. The Data Monitoring Committee saw a huge effect on a very important outcome, blindness. And what does it do? So what it did was it said, we cannot continue this study. We cannot allow this study to go on. And it was in the middle of recruitment, and people on the control arm were going blind, and people on the treatment arm were seeing. The endpoint it was a much more complicated endpoint. It wasn't just visual, but there was no question. And we went through imagining all kinds of extreme boundaries that could have been set up, and any one of them would have crossed. And so the DMC recommended stopping, infuriating the company because they said, you told us, we were told never to stop, angering the FDA, which said, we told you not to stop. But the study went to an AdCom, and the AdCom said, you have to approve this drug, and it was approved. So there is an example of a complete violation. So when you know the investigators, I think it's important to follow the guidelines, but don't be terrified by violating them. All right, so that was our experience with Rewind and with Cure. We knew the investigators very well. We knew how they thought. And so we were nervous about violating the guidelines, but not terrified. Understand what the investigators really want, because it may not be written clearly. They don't know what the data are actually going to be. So even when they haven't expressed their view fully. So as I said, for Cure and Rewind, the PIs were from population health sciences and Hamilton. We all knew them very well. We knew how they thought. And we suspected that they hadn't considered the scenarios we were seeing. So we suspected in Cure that they didn't realize there were going to be as many intracranial bleeds as we were seeing, and that they were going to all be in the treatment arm. And for Rewind, we thought they didn't think there's going to be no difference in women. So we put ourselves in their heads. We knew them well enough to do that. And so we were pretty comfortable, except we were worried about lawsuits, but not from them. But if you don't know the investigators, it's much more difficult. For the heart failure futility case, we didn't know the investigators. And by we, I'm saying the entire DSMB, we didn't know them. But we asked ourselves, what was the harm in continuing? The participants had experienced whatever risks they were going to have already. So if they had joined partly for altruistic reasons, what is the benefit in heart failure patients in general? They would want us to continue. And so we felt, well, the only reason to stop it was for the financial benefit of the company, and that was not our charge. For the 52-week study, that was more complicated because we really didn't, because people were still being recruited, and we didn't know what the company and the investigators, we didn't know how they thought. So the chair spoke to the chief medical officer and played out examples. What if this happens? What if that happens? What if the other happens? To get a sense of how the investigators were thinking. Because remember, when a charter is written or a protocol is written, you don't think about all the things that might happen. You think of what you think is likely to happen. And then something happens that's different, and you haven't thought that it's not surprising that those different things hadn't been in the investigator's mind. So in summaries, boundaries are guidelines. Generally, you should follow them. I don't want you to go home and say, oh, Janet says whenever you have boundaries, don't pay any attention. No, generally you should follow them because they've been thought out carefully. There's been a lot of reasons for them, but you're not a robot. Think about the consequences of violating your charge. What happens if you do something different from what your boundaries or guidelines tell you? How will recommending stopping or not stopping affect the participants? So again, with the heart failure case, it was easy. It wouldn't affect them. They would be treated just the same, whether the study stopped or didn't stop. In fact, they'd probably be treated better if the study continued because they'd be watched more carefully than if they had been just going off to their regular doctors. How would the sponsor respond? And how would the FDA respond? One of the things a committee doesn't want to do is recommend stopping the study early for overwhelming benefit, and then have the FDA say, well, sorry, we're not approving this. So one of the things that's really important when you're sitting on a DSMB or you're so difficult because we knew that the FDA had said, don't stop. And we felt that that had the probability of preventing approval of the drug. And yet the committee felt so strongly that it just couldn't bear to watch the harm in the control group. And finally, the DMC should operate by consensus, not by voting. And I've become believing that stronger and stronger as time goes on, because operating by consensus means you go around the room and you ask people to express their views, and you formulate your conclusion in a way that pulls together the views of everyone in a nuanced way. And if there's not consensus, then you continue, you do something else, but you aim for consensus and not just, you'll see in lots of charters, things like if there's a you have to have an odd number of members because that way there'll be a majority, or the chair breaks the tie. There should not be. That just means that upfront, the sponsor is saying, we don't care whether there's a majority or, we don't care whether the committee is operating as a whole. We're looking for votes, and I don't think that's healthy. And also, do not record meetings. The discussions in a closed session can be very, very emotional. They can express risk. They can express lack of confidence, all kinds of things. And when there are recordings, that can chill the conversation. And I've come to believe that even when somebody says, well, we record them just so we take good minutes and then we destroy the records. I don't believe that. Whether they do or not, we know from the Nixon tapes that you can find things on tapes recording even after it's supposedly been destroyed. So, and actually, we have a paper coming out saying this: no voting, don't record meetings, and don't record meetings, and in minutes, don't say who says what. This, if you're operating by consensus, it's a committee operating as a whole, and especially when this is especially important when it is violating the guidelines that has been given, that it is looked at. This is a committee as a whole who made those recommendations. And I want to say again, generally follow the guidelines. Do not take this as a, oh, go do what you want. Don't be a wild committee, but don't be a robot either. Thanks very much. Questions?
Thank you, Janet. I think there's one in the chat from Janet Holbrook, if you want to take a look. I can read it too.
Hi, Janet. Let's see. Oh, so Janet writes, for the last trial, the one without stopping margins by FDA direction, did they stop the trial? Yes, the trial was stopped. The committee recommended stopping, and with great, I was gonna say with great nervousness. No, they weren't nervous because they knew that it was the right thing to do, that they just could, that they felt it was unethical to continue to randomize participants when, I mean, the hazard ratio was like 0.2 with a confidence interval that was so narrow. So yes, they stopped the trial and the FDA did approve.
Doug has his hand raised as well.
So I think it's the rewind trial, but the one where the infect at the interim look was entirely in males and not in women. Did you consider stopping the trial in males and continuing in women?
Yes, we did consider that. That was one of the discussions we had. And the decision was that would be just too confusing and not to do that. But Doug, that's a really important question. And that's one of the reasons that I don't like things recorded, right? Because there was discussion about, is it ethical to, when we knew that the effect in men was so great, is it ethical to have them continue? And the decision was, well, if you believe that this is just that this may be a noisy thing coming from subgroups, then what you're seeing in the men is probably an overestimate of what's in the men, and what you're seeing in the women is probably an underestimate, and so it's okay to continue. What would have happened had the next meeting, had there been more meetings after that, and the next meeting had showed the same thing? I don't know what we would have done, but I was going to say we were comfortable. We ended up being comfortable with the decision to continue everybody.
So, I mean, it seems to me that there's probably a high likelihood that it could have been noise in the system, unless there was a good biologic reason to expect a sex-based difference, which, and I don't know the study well enough to know if there was a good biologic reason to expect that, but I think subgroups are often difficult to interpret because of the possibility of, as you said, noise in the system.
Yeah, there was a discussion of whether it was biologically plausible, and the answer from the physicians was they didn't see a reason for plausibility. And clearly from what the FDA did, that it didn't mention sex at all. It must not have believed that there was a biological plausibility.
And Dan Hanley has his hand up.
The example you gave from the CURE study of clopidogrel, where there was a consistent efficacy finding, and there was also an important safety finding of bleeding and intracranial bleeding, and you chose to continue, or the committee consensus was to continue. How did the continuation clarify the safety issue? And was it resolved at the end of the study?
So that's a really good question. I would say it didn't, there weren't that many more bleeds in the subsequent. Well, actually, let me start by saying something else. That, remember, the endpoint was cardiovascular death, non-fatal MI, and non-fatal stroke. I think that had the endpoint been dominated by cardiovascular death, we may have made a different decision. Because there were many fewer cardiovascular deaths than there were non-fatal MIs and non-fatal strokes. And then there were other deaths, and they were kind of evenly distributed. So we didn't feel we were balancing death against intracranial bleed. And as I say, I don't know what our decision would have been had the death difference been greater. But no, and actually the question you raised is a question that came up in the committee. So if the study continues, it didn't continue much longer, and there's not enough, and there aren't very many other bleeds, have we learned any? The decision was, or the thinking was, that there would be just more data to evaluate the risk of bleeds. At the end of the day, would the conclusion have been different had we stopped or not? I don't know.
Did the point estimate or the confidence intervals for any bleeding or serious brain bleeding change substantially?
Not substantially. It got a little tighter but there weren't that many subsequent bleeds. But it did give a sense of, and I don't know enough about the data, don't know enough about the propensity to bleed. It did give a sense of the timing of bleeds, because there was now more, it's not only more bleeds, but more time on drug.
And you had a full a priori sample size, which gave probably further confidence. Thank you very much. I think there's another question.
Yeah, we have one in the chat from Dave Shade. And if any students have, feel free to raise your hands as well.
I don't see the chat
The chat
question.
I can read it to you.
Okay.
So Dave Shade asked, let's see. When considering breaking the rules, do you think a DSMB would engage in discussion with the study team, i.e. the DCC, before making a final decision and recommendation? Or would this decision be discussed only in an executive session without participation of the study team, and then why?
Okay, that's a very complicated answer. So the committees don't like to talk to the study team very much, because if a committee is in this quandary and says, gee, we need to know what the study team would feel like, that's just unblinding the study, right? So they want to stay away from the study team. If you have a group that's not the study team, but that's higher up in whatever organization, sometimes the committee will go to that group and say, look, we've got this problem, we don't know how to deal with it. That's pretty rare, but sometimes that will happen. So going to somebody very high up to help make a decision. And then I forgot the next part of the question.
Sure. So the second part was, would this decision be discussed only in the executive session without participation of the
Yeah.
study team, and why?
Yeah, right. Right. Certainly without discussion of the study team, but there might be a discussion. All right. So here's another example where a committee didn't know what to do. We called in the CMO. So the case I described, the chair spoke to the CMO. Here we call the CMO, the chief medical officer, called the chief medical officer in, gave him a bunch of scenarios that could have happened, and asked him, none of which was the same as what we were seeing, but which collectively included what we were seeing. And then we would ask him to say what he would want and why. And then he went away, and then we were able to say, okay, we know, we can figure out what the committee, what the chief medical officer wants. Now we know how to think more. The case where the FDA had said no, and we actually did go to the no, don't stop. And we did go to the higher up people in the company, and they said, no, don't stop. The FDA said no. The decision of the committee was still, we are too uncomfortable seeing what's happening to the placebo patients. We're going to stop anyhow. That's the only time I've ever seen that happen. And there's a question here, a chat question about whether it's a violation of law or by the FDA. So I don't think there's any violation of law. The law doesn't cover this. And the FDA, in my experience, there isn't an FDA with respect to this. There are FDA divisions, and they're very different from each other. And even within the divisions, there are different reviewers. So in my experience, in almost every case, the FDA, I would say, was comfortable. I don't know. They went along with what the recommendation was from the DMC. And even in the case that I described, where we violated what the FDA said, the drug was eventually approved. And I hope it gets published. This study gets published. The description gets published, because I think it's really interesting.
All right, we're a little bit over time, but I think Jay has a comment or a question that he'd like to pose.
I have another question, if I can be
Thank
heard
you.
now.
Yeah.
I think I just got my audio back, and I'll try my video too. But like I said, I'm a little vacation lagged. I'm just curious about what your opinion is about the approval of the most recent Alzheimer's disease drug that was based on a phase 2B trial. Do you know what I'm speaking of? Like,
Yes, of course.
And it seems like because it was Bayesian, they didn't have one, to be frank, just one outcome for the trial, or a primary outcome, but it sort of didn't matter what happened with the primary outcome. They could base their decision, which to move forward might be okay, but it seems to approve on a secondary outcome. I just find it baffling.
Wait a minute, was that the Biogen one, or is it something else?
The second Biogen drug that has just been approved. It's lecanemab or lecanemab. Jay, can you say it correctly?
Oh, I can't.
Okay.
Correctly, but I know what you're talking about.
Yeah, no, I don't know. I don't know that case. I knew the previous one. I don't know that one.
Well, your comments on the previous one would be of interest to me too. I think I find this the kind of the Alzheimer's disease drug development process. It's like crash cars. You know, they can't do a trial without somehow screwing it up.
And I was appalled by the decision. And sorry, Janet. What?
So sorry for that comment. But they need you, Janet. You should go and contact them. How can they have two sets of clinical trials and screw them up so badly?
Yeah, no, I was appalled by the approval of the first, but I don't know about the second.
Oh, yeah. So in the first, it was that they had one good trial and one bad one, right?
Well,
And
it was more complicated than that, right? It was that there were two trials. The DSMB said stop for futility, but they had a stupid futility rule. And then, I mean, it's a much longer story to go into, but there was the statistician wrote the FDA statistician wrote a scathing review. The first advisory panel voted overwhelmingly no, and then the FDA brought them back and sort of made them vote yes. But then the payers wouldn't pay. I mean,
yeah, well, thank God for the payers.
yeah.
Anyway, yeah, well, yeah, the second one seems not quite as bad, but still just failed clinical trials that somehow get drugs approved. Failed, the first one for, as you said, not very good interim monitoring. They needed to break their rules. Exactly.
Yes, absolutely. And they needed to look at the data in a way that the rules didn't specify. One of the things that a lot of companies do is fail to give the ISRG all the data that they need. My view, again, this is to the students when you go out into the real world. If you're in a place that and you're dealing with a company and they will not give you the data, do not work with that company, because otherwise you'll be in the position of this Alzheimer's drug, where the first case, where they didn't have the data to be able to look behind the curtain.
All
Jay,
right.
it looks like you have a comment.
I did. I think, let me just say that for the second, the second drug, I think they looked very carefully at it because of the disaster with the first, and more closely than people normally would have looked at it. But that it was not my comment. I wanted to point out, Janet, that this is an excellent talk, and my comments are not at all scientific, as yours brilliantly were. But there are times the sponsors should overrule their boundaries. And one case was, I was on the DMC for a high-profile oncology trial. We had a scheduled, planned interim analysis. Everybody agreed to the dates. And the sponsor said, well, you can't meet on that date, because if you do, we would have to put the result of it in our quarterly report to shareholders. And since we don't know what you'll decide, you can't meet. And we said that this, for all the reasons you said that everybody on this Zoom knows, there are ethical issues in that. And we fought back and forth. They wouldn't change it, but they did. And they said, okay, but from now on, you will not be able to meet if it conflicts with a quarterly report. And we said, we'll worry about that later. But the other is, I'm on a trial now with a huge pharma company, a rare pediatric indication. We've been on this 3 years. Most patients, when they can actually find them, fail in the screening. And we have 8 patients after 3 years. If it's a good meeting, we add 1 new patient. Now, the sponsor is a prisoner of their SOPs on this. First, the woman physician who is in charge reads all decisions made by the sponsor since our last meeting and gives us links, a new policy on use of external data, a new policy on change of estimands. We're not going to do any of that on this trial, but their SOPs say she should make the announcement. Now, she did become a grandmother since our previous meeting. That was the only sensible announcement she made. And you know, the tables that these huge companies have, the table has a digit and the period, and then there's 7 more digits after that, and have to look very carefully at the last 2 or 3 digits to understand what the table is. So I said to the reporting statistician, in this case is an employee of the company, and but that's okay for this trial. But I said, okay, for the purpose of this committee, these tables, they have mostly zeros and ones in the columns. Can you at least renumber them? Table 1, table 2, table 3, just for this committee? And I can't change the numbers of these tables. We have to follow the SOPs. The final thing is there are 12 company employees that attend these meetings. And the same 12 would attend if there were 2,000 patients on this trial, because those are the SOPs. Couldn't they rotate and say, okay, Susan, I'll take it this time, you take it next time, or something like that? And those are not so terrible, but I think there's a time where they should realize with a trial like this, there may not even be a new patient. Why do they all have to be present? Anyway, I just thought I'd add that.
Yeah, people get to be prisoners of rules and of SOPs.
Thank you. We are way over time. Thank you so much, Janet, for being so generous with it for us. And I guess that's it for today. So thank you all for joining, and